Assalamu alaikum and welcome everyone to the second part of Molecular Biology ASCP preparation series, Molecular Techniques. I'm Dr. Lai Abdelati and I'm really excited to share my knowledge and experiences with all of you through this series. So let's dive in together. Our focus for this season will be on nucleic acid purification, a cornerstone of molecular diagnostic and a high-yield topic for Molecular Biology ASCP exam. So let's dive in. Alright, let's define what is the DNA purification before we get into the questions. Actually, nucleic acid purification is the fundamental techniques in molecular biology that allow scientists to extract DNA or RNA from various biological samples such as the cell, tissue, blood, and environmental specimens. This process is essential for applications like PCR, sequencing, gene editing, and diagnostic, where the purity of nucleic acid directly affects the experimental success. It involves three key steps, starting from lysis, separation, and recovery. In lysis step, where the cell or vital particle are broken open to release nucleic acids, then we go to separation step, which remove contaminant like protein, lipids, and carbohydrates. And the final step is recovery. In recovery, where the purified nucleic acids are concentrated and supplied for future use. All right? The common purification methods include chemical extraction, solid phase extraction, and enzymatic treatment. In chemical extraction, using phenol chloroform for protein removal. In the solid phase extraction, with the silica column or magnetic beads under specific salt and pH condition. And lastly, in the enzymatic treatment, using RNase or proteinase K to break down unwanted molecule. All right? All right, now let's move to the question part and see the first question we have today. All right, which of the following is the primary purpose of nucleic acid purification? The primary purpose of nucleic acid purification is to isolate nucleic acid, even DNA or RNA, from other cellular components and contaminants. All right? All right. Which of the following is a key advantage of using silica-based columns for nucleic acid purification? All right, the correct answer is rapid processing time. As we know, silica-based column allow for rapid and efficient purification of nucleic acids, so making them a popular choice in many protocols. All right. All right, the next question here. Which of the following methods is commonly used to disrupt cells during nucleic acid extraction? The correct answer is sonication. Actually, sonication is a widely used technique in nucleic acid extraction. It works by using ultrasonic waves to break open cell membrane, allowing nucleic acid inside to be released. All right? Okay, next question here. Which of the following is the common contaminant in nucleic acid preparations that can inhibit downstream application? All right. The correct answer is all of the above. Actually, protein, lipids, and carbohydrates are common contaminants that can interfere with the nucleic acid purity and downstream application. So these unwanted elements need to be removed to ensure a clean and functional sample. All right, let's move on. The next question. Which of the following is the common method for concentrating nucleic acids after purification? Right, the correct answer is lyophilization. Actually, lyophilization, also called freeze drying, is the method used to concentrate nucleic acid. So this process removes water from the sample by applying low temperature and pressure, which help preserve the nucleic acid integrity. It ensures that the sample remains stable, making it easier to store or use for further analysis. All right? Right, the question here, what is the function of RNAs in DNA purification protocols? The correct answer is to degrade RNA. Actually, RNase is the enzyme that degrades RNA, so ensuring that DNA preparation are free from RNA contamination. All right, during nucleic acid purification, what is the purpose of using lysis buffer? The correct answer is to dissolve cell membranes and release nucleic acids. Actually, this lysis buffer contain detergent and other chemicals that break down cell membranes, releasing nucleic acids into solutions. Okay, let's see the next question here. Which of the following reagents is commonly used to remove proteins during nucleic acid purification? The correct answer is phenol. Phenol is used in phenol chloroform extraction to denature and remove proteins from nucleic acid preparations. So, so I made a sign for P in protein and P in phenol to remember protein is removed by phenol. All right, let's move on. All right, in phenol chloroform extraction, what is the purpose of adding chloroform? The correct answer to separate proteins from nucleic acids. Actually, as we know, chloroform helps to separate proteins from nucleic acids by creating a distinct phase where proteins are denatured and separated from the aqueous phase containing nucleic acids. All right? Okay, let's see the next question here. What is the purpose of using chautropic agent in nucleic acid purification? The correct answer is to denature proteins and nucleic acid. Actually, the purpose of using chautropic agents in nucleic acid purification is to disrupt the structure of proteins and nucleic acids, making it easier to separate and purify them. Okay, let's move on. Okay, let's see the next question here. Which of the following is the common indicator of RNA integrity in purified sample? RNA integrity, okay, not RNA ratio. All right. The integrity of RNA can be evaluated by observing the band pattern producing during the electrophoresis. So when RNA is intact, distinct bands appear corresponding to the ribosomal RNA as we see in the image here. Okay? If you found this content helpful, please don't forget to like, share, and subscribe to our channel. Your supports mean the world to me and help me create more valuable content for you. Thank you for being part of this journey. Let's continue. All right. What is the primary purpose of adding guanidinium thiocyanate in RNA purification? The correct answer here is to denature proteins and inhibit RNAs. All right. Actually, guanidinium thiocyanate is considered as the chautropic agent that disrupts protein structure, inactivates RNAs, and help release nucleic acid from cells. All right. All right. The question here: which component is the critical in DNA storage buffers to prevent degradation? The correct answer is EDTA. EDTA is chelating agent that bind to magnesium ion, which are essential for the activity of DNAs. We know DNAs. DNAs is enzymes that break down DNA. Okay? So by removing magnesium, EDTA prevents these enzymes from functioning, thereby protecting DNA from degradation. All right? Let's move on. All right. All right. Next question. What is the role of ethanol in nucleic acid purification? All right, the correct answer is to precipitate nucleic acids. Okay, actually, ethanol is used to precipitate nucleic. All right, next question here. Why is 70% ethanol used in DNA precipitation? The correct answer is to remove salt without dissolving DNA. Actually, 70% ethanol effectively washes away salt while keeping DNA insoluble. Okay, what happens if we use the 100% ethanol? Actually, higher ethanol concentration can excessively dehydrate the DNA, so making it harder to redissolve in water or buffer solution for further use. All right. So here, why we use 70% ethanol instead of 100% ethanol. All right. Let's move on. All right. The next question here: what is the role of beta-mercaptoethanol in RNA isolation? The correct answer is inactivate RNases by reducing disulfide bonds. As we know, beta-mercaptoethanol is a reducing agent that disrupts RNase activity by breaking disulfide bonds in these enzymes. Okay, let's move on. All right, next question. Magnetic bead-based nucleic acid purification relies on. The correct answer is binding under high salt conditions. All right. All right. In the presence of high salt, nucleic acid bind strongly to the surface of magnetic beads, while unwanted cellular components such as the protein and lipids are left behind. Okay. The beads now carrying the nucleic acid are thoroughly washed to remove any remaining impurities. Then a magnetic field is then applied to separate the beads from the wash solution, ensuring that only the purified nucleic acid remain. Finally, a low salt or water-based solution is used to release the nucleic acids from the beads, making them ready for downstream application. All right, let's move on. Okay, what is the purpose of proteinase K in DNA extraction? The correct answer is degrade cellular protein. From the name proteinase K, it means something related to the protein. Okay, So the correct answer is degraded cellular proteins. Let's move on. Let's see the next question. Which step remove lipids during genomic DNA extraction from tissues? The correct answer is organic extraction, I mean phenol chloroform extraction. Okay? As we know, DNA or RNA is hydrophilic, right? Which means it dissolves in aqueous layer during phenol chloroform extraction. This aqueous layer, please focus on the image here. This aqueous layer is found at the top of the tube after centrifugation. Okay? Protein, on the other hand, typically collected at the interface between the aqueous and organic layers, forming a visible boundary, as we see in the image here. Lipids, being hydrophobic, do not mix with the aqueous layer. Instead, they separate and end up in the organic, I mean phenol chloroform layer, which is the lower phase. Okay? Let's move on. All right, the next question. What is the purpose of isopropanol in nucleic acid purification? The correct answer is precipitate nucleic acid at room temperature. Actually, when isopropanol is added to the DNA or RNA solution, it disrupts the hydration shell surrounding the nucleic acid molecules. This disruption causes the nucleic acid to aggregate and form precipitate, as we see in the image here, which can be easily pellet through the centrifugation. All right, let's move on. Okay, the next question here. Which step in plasmid purification removes chromosomal DNA? All right, the correct answer is alkaline lysis and centrifugation. Actually, alkaline lysis denatures chromosomal DNA, which is removed via centrifugation, leaving only plasmids in the solutions. All right, let's move on. Okay, the next question here. Lysozyme is used in bacterial DNA extraction too. The correct answer is digest peptidoglycan in cell wall. Actually, lysozymes plays a critical role in breaking down bacterial cell wall. It does this by hydrolyzing peptidoglycan, which is the structural component that gives bacteria their rigidity and shape. All right, let's move on. All right, which method is best for concentrating diluted DNA? The correct answer is ethanol precipitation. Actually, by adding ethanol to the DNA solution, the solubility of DNA is reduced, causing it to aggregate and precipitate, as we see in the image here. All right, let's move on. Okay, in the context of nucleic acid purification, what does the term elution refer to? All right. Elution is the process of separating and transferring nucleic acid from solid phase like silica column into a solutions. Okay. So the correct answer here is the release of nucleic acid from a solid phase. Let's move on. Okay. Next question. In column-based DNA purification, the elution buffer typically has which one of them? All right. In column-based DNA purification, Elution is the final step where DNA is released from the silica column. So this is achieved by using elution buffer with low salt concentration and neutral pH. So this buffer disrupts the bond between the DNA and silica, so allowing the DNA to dissolve in the solution for collection. All right? So the correct answer here is low salt and neutral pH. All right. Which of the following is the common method for assessing the purity of nucleic acid samples? The correct answer is spectrophotometry. Okay? Actually, this technique is commonly used to assess the purity and concentration of nucleic acid samples by measuring absorbance at specific wavelengths. All right, next question here. What is the typical absorbance ratio, 260 over 280, indicating pure DNA? All right, the correct answer is 1.8. Actually, a ratio of 1.8 at absorbance 260 over 280 is generally considered indicative of pure DNA with minimal protein contamination. All right? All right. The question here, a low absorbance 260 over 280 ratio, example 1.2, in a DNA sample suggested contamination with. All right. When a DNA sample, okay, has a low absorbance, 260 over 280 ratio, I mean below the expected range of 1.8 for pure DNA, it suggests that the presence of protein contamination or other substance like phenol. Okay? All right. What does a high absorbance 260 over 280 ratio more than 2 suggest? All right, the correct answer is RNA contamination. Okay, actually RNA typically has absorbance over 260 over 280 ratio of around 2. Okay, while pure DNA has a ratio close to the 1.8, as I said. Okay, so if the ratio exceeds 2 in DNA sample, it suggests that RNA contamination. Okay, this distinction is critical for ensuring the purity of DNA samples during analysis. Okay, next question. If a DNA sample has an absorbance 260 over 230 ratio, let's focus, okay? Less than 1.8, it's likely contaminated with. The correct answer is carbohydrates or phenol, okay? Let's move on. All right, the last slide we have today highlights two critical ratios used to assess the purity of nucleic acid, DNA and RNA using spectrophotometry. The absorbance 260 over 280 ratio and 260 over 230 ratio. Let's start from the first one, absorbance 260 over 280 ratio. This is used to detect contamination from protein or phenol. Okay, contamination from protein or phenol. And pure DNA typically has a ratio around 1.8, while pure RNA shows a ratio closer to 2. Okay, so if the ratio is lower than expected, it suggests protein or phenol contamination. Okay, this is the first point. The second point, absorbance 260 over 230 ratio. This evaluate other type of contaminants such as the carbohydrates, residual phenol, guanidine, or salts. Okay, the ideal range of pure nucleic acid is around 2 to 2.2. All right, a lower ratio here indicate the presence of these impurities. As I said, phenol, guanidine, carbohydrates, and salts. All right. So by using both ratio, we get a comprehensive view of nucleic acid purity, ensuring that our samples meet the standard required for downstream application. All right, that's all for today. I hope you enjoyed this lecture. Please don't forget to like, comment if you have any questions, share and subscribe to our channel to receive all the latest lecture. I also encourage everyone taking the test to share your question with us so that everyone can benefit. Salam.